Chapter 3, Protein Structure and Function. Proteins are one of the 4 macromolecules that we'll be discussing in this class, and proteins are the most abundant and versatile macromolecules in life. Proteins are made of amino acids, and amino acids are considered the building blocks of proteins. There are 20 amino acids. Now, because proteins are so diverse, their structures are also diverse. So we'll talk about 4 levels of structure: primary, secondary, tertiary, and quaternary. Then we'll talk about protein function, and lastly, we'll talk about the diverse role of proteins in living cells. Okay, so were proteins responsible for the origin of life? I'm going to beat to the chase and I'm going to say no. Are proteins extremely important? Yes, they are, but they did not trigger life itself or the very first cell. Amino acids are the building blocks of proteins, and you'll need to know this for the next test. In Stanley Miller's experiment, where we saw that in the previous chapter of the atmosphere and the ocean in bulbs of glass, we know that under certain conditions, we can in fact synthesize amino acids under lab conditions and reflect those of primordial Earth. So as I mentioned before, proteins probably did not spark life. Because to spark life, you need to have information, replication, and evolution. And information and replication is typically found on DNA, not proteins. Okay, so if we look at the structure of an amino acid, which is the basic building block of a protein, we see that there's a central carbon. And coming out from this central carbon is a hydrogen. There's an amino group, NH2, a carboxyl group, COOH, and then an R group. This R group represents side chain, and this is what makes each of the 20 amino acids unique. I love this drawing here of the non-ionized form of an amino acid. Notice the backbone is NCC. And from that central carbon, you see that there's an amino group, on the other side a carboxyl group, above a hydrogen, and below the side chain. And keep in mind, it's the side chain that makes each of the 20 amino acids unique. The combination of amino and carboxyl functional groups are key to how the molecules behave. And in this particular case, amino and carboxyl groups easily ionize. Remember, when you ionize an atom, there's either a loss or gain of electron. When there's a loss or gain of electron, that means they become charged. If by chance they become charged, then water is charged also. And remember, birds of a feather flock together. So if they become charged and water's charged, then they're very reactive in a water solution. In the ionized form, you'll see that the amino group gains electrons and a proton, whereas in the carboxyl side, there's a loss of a proton or a hydrogen and electrons. The nature of the side chains. As I mentioned earlier, the R group is what gives a character to each of the 20 amino acids. And each of these properties in the side chains vary from one amino acid to the next. And these R groups can participate in chemical reactions. And then some side chains may have functional groups, but others may not. If you look in your book at figure 3.2, you see that there's a listing of all of the 20 amino acids, and you see the R group or the side chain, and you see how it makes each of the 20 amino acids unique. So if we were to put them into groups or clusters, we would look at their solubility, their affinity towards water, whether they're hydrophilic or whether they're hydrophobic. So if they go into a group, there's 3 basic questions that we ask. Does the side chain have a negative charge? Does the side chain have a positive charge? Is the side chain uncharged? And that will help you understand the different types of amino acids. For your upcoming test, you don't have to know the charge of the side chains. Amino acids polymerize when they form bonds between one carboxyl group of one amino acid and an amino group of another one. So that bond that forms between carbon and nitrogen is called a peptide bond. And this bond is important, and you will have to know it for your upcoming tests. I love this illustration right here because it actually shows that there is a dehydration reaction, and you can see it right here in those that are have the blue blocks around them. H and O is water, and in a condensation reaction, water goes out. A peptide bond is formed between carbon and nitrogen, and here you have a peptide bond. There are three key points about peptide bonds. First is there's R group orientations. The side chains extend out when one amino acid starts attaching to another. And because of this, they can start reacting with each other. There's also directionality of a protein. In fact, one side is the nitrogen terminus, which has the amino group, and the other side is the carbon terminus, which has the carboxyl group. The third important point when we start stringing amino acids together is that all of a sudden there's a lot of flexibility. It may not be in the peptide bond itself, but it's in the single bonds next to the peptide bond. And that's why proteins have such diverse roles and diverse shapes. Here's an example of a string of amino acids that are forming a protein. Notice the backbone, NCC, 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 NCC. And each of these amino acids are attached by a peptide bond through a condensation reaction. Notice one side has a nitrogen terminus and ends in an amino group, and the other side is the carbon terminus ending in a carboxyl group. This figure 3.5 shows the flexibility of a string of amino acids forming a protein. So once peptide bonds are formed, we can have oligopeptides, polypeptides, and proteins. I'm going to be honest with you, I hardly ever hear the term oligopeptides, but I definitely hear the term polypeptides and proteins, and in your book they're used interchangeably. So polypeptide means protein and vice versa. Can amino acids spontaneously assemble into proteins? And researchers have been able to generate stable polymers in lab using free amino acids. They observed that amino acids being formed and even polymerized under hot metal-rich conditions. But it's still unclear actually how proteins formed under early Earth conditions. What do proteins look like? Well, we already know that there's directionality and there's flexibility. And because of that, there's a diversity of shape and of course chemical properties. So structures give rise to different functions of proteins. In figure 3.6, you can see the different types and shapes of a protein. Notice up at the top is collagen. And isn't it true, like for those of you who buy creams, you can buy creams that have collagen in them. And collagen actually looks like strands of hair. And then down below, you see a Tata box, which we'll talk about later, porins and chymotrypsin. And all of these look different, so there's great diversity among protein shapes and of course their function. So because of their diversity, we need to talk about structure. And proteins have structure on 4 different levels. So the first level of structure is the primary structure. And the primary structure indicates the unique sequence of amino acids. It's just as if I had brought beads to class and I'm asking each student in class to make a necklace. The way that you string them together is going to give distinction to your necklace. The same thing is true of a protein. The way the amino acids are strung together will give the uniqueness of the final protein. So keep in mind there's 20 types of amino acids. To me, I almost liken it to the number of letters in an alphabet. And from there you can make so many words, and those words together make so many unique sentences. The primary structure is fundamental to higher levels of protein structure, and this could definitely be a test question. In other words, that primary structure, well, the secondary, tertiary, and quaternary levels are dependent on how amino acids are sequenced at the very beginning. Amino acid R groups are affected by the properties and function of each of the amino acids. And so the way that they are strung together, again, is very important. Here's a classic example of when there's a mistake. In the frame up above is the normal sequence of hemoglobin. And so notice red blood cells have that traditional like little red frisbee shape. But if there's one change in only one amino acid, notice we get a sickle cell. And this is what causes sickle cell anemia. So instead of glucine, we have valine, and you can see the tremendous change in shape of a red blood cell. The secondary structure. The secondary structure is the beginning of the three-dimensional shape of a protein. And what holds this shape together is hydrogen bonds. You know, this could be a good test question. In the secondary structure, how is that three-dimensional shape held together? It's held together by hydrogen bonds. And so there's 2 basic shapes. And you know, I liken these shapes to actually hairstyles. The alpha helix is like Shirley Temple's curls, and the beta pleated sheets are like Bart Simpson's hair. So in figure 3.8, you can see that this is the first time that proteins are getting their three-dimensional shape. It's either going to be like a ringlet or it's going to be corrugated. But nonetheless, in both shapes, hydrogen bonds are holding those shapes together. So here you see the 2 basic shapes, an alpha helix and a beta pleated sheet. And you see that this is how they're represented in your book. So here are some ways of seeing an alpha helix and a beta pleated sheet. The third level is the tertiary structure, and this results from interactions between the R groups. So these cause the backbone of the protein to bend and to fold, and it further gives shape to this three-dimensional structure. So what are the ways that R groups can interact and form ways that keep the three-dimensional structure of a protein? Well, one is hydrogen bonding. Number 2 is hydrophobic interactions. 3 is van der Waals interactions. 4 is covalent bonding. And 5 is ionic bonding. If you look at all of these type of interactions, you are already familiar with all of them except for number 3. Number 3 is the van der Waals interactions. And this refers to weak electrical interactions between hydrophobic side chains. So you already know that hydrophobic is water hating. So these are chains that actually share their electrons equally. I really appreciate figure 3.9 because it shows the 5 ways that side chains interact to give more dimension to a protein. And you know, more than likely, this will also be a test question. So this diagram is really wonderful. The quaternary structure is the last structure, and what it's doing is it's forming these little macromolecular machines, and these macromolecular machines are made out of different units of polypeptides, or shall we say small proteins. So there's multiple polypeptides, and they can either be dimers that are homodimers or heterodimers. And you definitely don't need to know that for the test, but you do need to know that the quaternary structure is made up of multiple units of polypeptides or proteins. Here are some examples of tertiary structures. So notice here you see multiple alpha helices or beta pleated sheets, or actually in the lower left-hand or right-hand corner, you see a combination of both. So as I mentioned before, if we have multiple polypeptides working together, they're often referred to as macromolecular machines. And these proteins carry out particular functions within a cell. The protein structure is hierarchical. In other words, the primary structure comes first, then the secondary, then tertiary, and then quaternary. And remember, the primary structure is the most fundamental structure. All other levels are dependent on this primary structure of the sequence of amino acids. In the quaternary structure, the way that a protein folds is very important. So there's different levels of structure responsible for the diversity of proteins. And protein folding is directed by the sequence of amino acids. In table 3.1, it gives a summary of all the levels of hierarchical structure of protein. I love this table. If you can look at this table in a glance and you understand most of what is being mentioned here, you really have understood most of my lecture. Here are some examples of macromolecular machines, and here you see the CRO protein, which is a dimer, and then hemoglobin, which we see in our red blood cells. Normal folding, protein folding, is crucial, and it is often spontaneous. And this is a result of chemical bonds and interactions that occur among R groups. And folded molecule is more energetically stable than an unfolded molecule. So when a protein isn't folded, it has less potential energy. When we say that a protein is denatured and not functioning anymore, that is because it has been unfolded and it does not have its proper function. So what helps the molecules fold? Before I go on, is you intuitively know that the folding is important. I remember one of my children, when they were really young, they always had a temperature, and really before they were 1 year old, they were in a tepid bed most of the time. Why was that? Why was I so concerned about high temperature? And it's because proteins are very sensitive to extremes in temperature. So I did not want any of their proteins, my child's protein, to become denatured. So here we see that folding is also helped by these other proteins called molecular chaperones. And they're responsible for blocking inappropriate interactions between unfolded proteins. And so an example in your book is the heat shock protein HSP90. And here on the left-hand side, you see a properly folded protein, and on the other side, it's when it's denatured and unfolded. Each protein has a characteristic folded shape necessary for its function. So notice here that the continuous theme is that shape is important for function. Many proteins are flexible and dynamic when in an inactive state. Each protein will exist in a variety of shapes until prompted to adopt a folded and functional forms. So shape changing is very common among proteins. And in fact, that can be a trigger to turn a chemical reaction on or to trigger it off. So protein folding is regulated and shape changing is very common, and we'll talk about it in future chapters. Can folding be infectious? And unfortunately, the answer is yes. There's something called a prion, and a prion is a misfolded protein. And the thing about prions, not only is it misfolded, but it can get other proteins to also become misfolded and change their shape. And one disease caused by prions is the mad cow disease. A couple of, I'm going to say decades ago, we did not purchase any meat from Europe, and it's because of an outbreak of mad cow disease actually in England. And what had happened there is that dairy cows and meat cows were actually being fed the carcasses of other herbivores, which I know is inappropriate and sort of unnatural, but that was the spread of mad cow disease. And it says prion infectivity is linked to structure. So notice here up on the left-hand side, you see this is the way that it should be properly folded. It looks like 4 alpha helices, but a prion can make it refold. And here you see a combination of alpha helices and beta pleated sheets. So why are proteins so important in the cell? And as you can see by this list here, is that they are involved in many functions within the cell. One is catalysis, which is speeding up chemical reactions. Another is structure. Not only do we think of muscle and protein, but also the shape of individual cells. Proteins are important for movement. They're motor proteins that help expand and contract filaments. And proteins are also used for signaling between cells. There's the transport. The transport of molecules in and out and within a cell are often, those are involved with proteins. And then lastly, proteins are involved with defense. They're the antibodies that attack pathogens. Why are enzymes good catalysts? So keep in mind that when we're talking about proteins, one particular group of proteins are enzymes. So that is to say, all enzymes are proteins. So that makes them macromolecules. And these enzymes, these huge macromolecules, can catalyze reactions. And what they do is they hold reactants or a substrate in the active site. So a classic example of what an active site is, it's sort of where you put your key in an old-fashioned lock. That hole, that space would be the active site. So the reactants or the substrates fit in that active site. And in this example here, we see an enzyme, and that's indicated by the green structure. And the substrates or the reactants are what you see in red and blue. So they fit in this active site, and actually the bonds are stressed and the molecules are oriented in such a way that they can react with one another. Is it possible that a protein catalyst was the first molecule capable of replication? Researchers have shown amino acids could have polymerized to form small proteins, yet attempts to simulate this for the origin of life with proteins has not been successful. So, in other words, actually the formation of self-replicating enzymes has not been able to be synthesized under lab conditions. This is the end of the protein chapter. Thank you.